You're watching CTOM TV. Knowledge is power.
Well, I think a bit of a primer on coronaviruses would be a great place to start.
Okay.
So coronaviruses were first discovered in the 1950s.
Right.
They're referred to as the order Nidovirales.
All right.
And they first found them in chickens
Hmm.
that had infectious bronchitis. So bronchitis is not normally infectious, but this is a particular strain of infectious bronchitis that would wipe out these poultry farms. And if one chicken got it, almost all of the chickens got it. So literally, they would just have to kill all of the chickens and then spray down, the stalls and start from scratch.
Wow.
Years later, they started seeing it in other animals such as pigs and cows.
But we're going back to the 50s, you're
50s,
saying?
yeah,
To-
1950s,
okay.
yeah. And what we know now is that coronaviruses have evolved with bats. Bats are the perfect vector, and bats carry, different types of coronaviruses. And bats are the perfect transportation vector because they are mammals, but they also fly.
So they get around a lot.
They get around a lot, but they're also mammals, so they
Right.
share a lot of the features that
That we
humans
have.
have. That we have.
Unlike
Primates,
birds.
unlike birds, exactly.
Right.
So they're a flying mammal, and SARS, which is SARS-1, COVID-19 is SARS-2, but SARS-1 originally developed, they believe, in civets and potentially in pangolins, which is a type of anteater. Another form, Middle Eastern respiratory syndrome, was MERS, and that went from bats to camels. And the death rate for that was 37%. So it was quite scary. So
Shoot.
yeah, so from bats to camels to humans.
And I know there was a MERS outbreak in Asia,
South Korea,
2015?
Was it South Korea? Yeah.
Among a few other places. So that's not so long ago.
Right. So coronaviruses have a long history with humans. We have a complex relationship. So if you'd like to put on your Facebook profile of your relationship with coronavirus, you could put complicated. It's complicated.
Long-term,
it's complicated. So there's at least 4 different strains of coronaviruses that cause about 30% of the common colds that we get every year. So every year we're all infected with these 4 different strains, or the vast majority of us are. However, they typically just affect the upper respiratory tract and the sinus and the throat. Whereas the SARS subtypes and the MERS, they affect the lower respiratory tract in the lungs, and that's what makes them so deadly. So, literally, why is COVID-19 so deadly? Well, it's because it has the infectiousness of a regular coronavirus that we normally see that causes colds, that normally only causes the problems in the upper respiratory tract, but it has, the transmission, it's easily transmitted, but it has the deadly effect of infecting the lungs, such as SARS and MERS do, SARS-1 and MERS. So one of the reasons for this is it has something called a spike protein. All coronaviruses have spike proteins. But this particular version, COVID-19, the spike protein has a talent for binding and fusing with lung cells due to something called the ACE2 receptor. Okay. So the ACE2 receptor is a receptor in our cells. It's common in lung cells. It's in kidney cells and some other organs. But it provides a perfect opportunity for the COVID-19 to bind with and attach to lung cells, because it's a protein that's commonly expressed in lungs. So SARS-1, the original SARS, binded well to the ACE2 receptor. However, COVID-19 binds with 10 times more efficiency. So it's very, very effective, 10 times more effective at binding to this ACE2 in the lung tissue.
The lungs.
Yes. Now, unlike SARS and MERS, COVID-19 also uses a widely available enzyme called a furin. Now, this is a common enzyme in humans. Now, in a bit of a background, in order for the coronavirus to infect a cell, first off, what happens is the spike protein binds with the ACE2 receptor, and then it has to cleave it off at the stem of the cap, or the protein's cap from its stem. It needs to be cleaved off. And once that happens, it allows the membrane of the virus to bind with the membrane of the cell, and then it can spill its contents, the RNA, into the cell and infect it. So without that process, it's not able to do that.
I see.
So because furin is available in the lungs and is a common human protein, COVID-19 is adapted to use that, to cleave it. So it's able to infect us for a variety of different reasons. Number one, it's higher affinity for the ACE2. And number two, its ability to use the furin to complete the next step. Now, once the virus is infected in our cells, then typically it uses our cellular machinery to copy itself until it reaches a certain number and it spreads out. Now, most RNA viruses, and this is an RNA virus, most RNA viruses like this typically have very poor replication abilities. And that is because they have a high mutation rate. So in other words, a lot of the progenitor viral particles will be, not viable because they're so mutated from the original one. Coronaviruses are unique in that sense, is they have something called a proofreading function. And so what happens is, unlike most RNA viruses, they're able to fix their DNA or their RNA. So this is something that humans have too. When we reproduce, whenever we get damage to our DNA, we have a series of different proteins and enzymes called DNA repair enzymes that will fix it and will put a checkpoint on it and prevent that cell from copying itself and producing daughter cells until that DNA is perfect and it's all fixed.
Got it.
So coronaviruses are able to do that too with their RNA. So they're very unique. In case of RNA viruses, they're very unique. They've probably developed and evolved this proofreading function because they have a very large genome for an RNA virus. So RNA viruses are usually not that large, but coronavirus has about 30,000 nucleotides, which is a fairly large RNA virus. And this overall allows it to remain stable in its host. This combination of having many different genes and proofreading function is it allows it to remain stable in the host when there is no selective pressure. It doesn't need to mutate. It doesn't need to change. But when it needs to, it can rapidly evolve and adapt to new species.
Hence,
Exactly.
That's what we're seeing here.
So one of the most important aspects of viral transmission is its ability to shed. And that is how its host releases the viral particles to infect other hosts. So viruses replicate in order to shed and shed from its host. And then they shed their viral particles in mucus and snot and phlegm and even in breath. On molecules in your breath. So COVID-19 is a super shedder specifically in people that don't have symptoms. So in other words, when you get infected, if you don't have any symptoms, you are 55% more likely to be a super shedder. Or in fact, if you have mild symptoms, you are 55% more contagious as people with severe cases of it.
And why is that? How does that?
We don't know yet.
We don't know.
We're not sure. We just know that the symptomless people or the people with mild symptoms are super infectious. And importantly, in severe cases of people that require hospitalization on respirators and so on, in those patients, the virus can be still shed from those patients for up to 40 days after they've been infected. So, this is kind of an important thing that we're going to cover. So to give you some background on this, 100 million coronaviruses can fit on the head of a pin. It only takes about 1,000 to 10,000 particles to cause an infection. And they can live on, COVID-19 can live on air droplets for up to 3 hours.
Wow.
Some people are super infectors.
Right.
We haven't quite identified who and why.
Right.
We know that having low symptoms when you're infected means you're probably shedding a lot. So the bottom line is we need to test everyone, and that's going to be very, very important.
Thank you for watching Ketome TV. Subscribe below and stay informed.